Very good afternoon. Welcome to the Technology Showcase on Process Innovations in Pharmaceuticals. The agenda for today's talk will be for today's showcase will be as follows. So we have introduction to Venture Center and TechX first. After that, there will be a very brief introduction about the matchmaker and the showcase, followed by the technology pitches, which all of you all are eagerly waiting for, which will be, the pitch will be for 5 minutes, and the scientist will be talking about their technology for 3 minutes, followed by discussion and Q&A session for 7 minutes. And this will be ending with the concluding remarks by the advisors. So with this agenda in mind, we will now begin with a very brief introduction of Venture Center and TechX.
Good afternoon. We welcome all the participants to the Technology Showcase event on Process Innovations in Pharmaceuticals. We have 7 outstanding technologies lined up for the showcase. Before we proceed with our presentations for the day, let me give you a brief introduction about the organizers of the event. Venture Center is India's award-winning technology business incubator for science and deep tech startups. Since our inception in 2007, marking 18 years of innovation, we have nurtured over 200 startups, empowering breakthrough ideas with a full spectrum support system. At Venture Center, our mission is to catalyze inventive ventures that tackle the world's most pressing challenges using science and technology. Our ecosystem is built on 6 key pillars, from new venture creation and early stage funding to dynamic knowledge sharing, business incubation, innovation management, and fostering a robust innovation ecosystem. Our state-of-the-art resource centers serve as hubs of excellence, providing cutting-edge infrastructure, expert mentoring, and knowledge sharing, business incubation, innovation management, and fostering a robust innovation ecosystem. Our state-of-the-art resource centers serve as hubs of excellence, providing cutting-edge infrastructure, expert mentoring, and specialized facilities to accelerate our startup journey. TechX is the vertical of Venture Center. It is a regional technology transfer hub, which is supported by MBM and BIRAC. We provide various services which help innovators and technology commercialization entities with tech transfer and IP support services. With this very brief introduction, I now hand over the forum to my colleague Pradnya Arathe, who will be setting the stage for this matchmaking.
I would now request Vedang to play a short video describing the matchmaker and the showcases. Please go ahead, Vedang.
Good afternoon all. Welcome to the Technology Showcase on Process Innovations in Pharmaceuticals, which is a pro bono effort of our team. Our technology matchmakers are aimed at theme-based matchmaking between technology providers and technology seekers. On one hand, we have technology providers from publicly funded R&D labs, academic institutes, and sometimes startups. On the other hand, we have technology seekers from industry, startups, entrepreneurs, and investors. The intended outcomes of the matchmakers is to have technology licensing deals, co-development agreements, sponsored research projects, spin-out, startups, new venture creation, etc. Today we are conducting 6th matchmaker and 9th technology showcase. All our matchmakers are managed through dedicated websites that we create for each matchmaker. On the left hand side of the screen, you can see the link for today's matchmaker and the snapshot of the website. We also create various marketing collaterals to publicize the matchmakers and the showcase and the pitches. On the right hand side, you can see a sample of the flyer that we have created. Let us take you all through the process of matchmaker. It starts with identifying a theme and putting together a group of advisors who are accomplished in their fields, domain experts having industry exposure. Then our team announces a call for calling technology submissions from the providers. We publicize this widely. At the same time, we align industry people about the upcoming technology showcase. Once the proposals are received, we seek guidance from our advisors to shortlist the technologies and curate the pitches. Our team works with these scientists to create the technology pitches, and the showcase day arrives. For today's showcase, we received more than 20 submissions. We had reached out to more than 30 institutes and more than 30 industries. We reached out for the showcase. The intended outcome of the showcase day is to have expression of interest from the industry. Once the expression of interest is received, our team then does the required lead management and necessary agreements. Today we have participation from two institutes, CSIR NCL Pune and CSIR CDRI Lucknow. On the technology seeker side, we are expecting representation from various pharma industries like Alchem, Cadila, Dr. Reddy's and many more. Few of them have requested anonymity. For this particular matchmaker, we have received guidance from very accomplished experts in their field who are from pharma industry. They have collective experience in science, technology, industry connects, projects, commercialization. Today we have with us Dr. Girij Pal Singh, who is Chief Technology Officer, Aquafarm Chemicals Private Limited. He has previously worked with Lupin. A very senior pharma professional, Dr. Gopakumar Nair, Designated Partner, Nanlex Associates LLP. Dr. Premnath, Head NCL Innovations, CSIR NCL and Director Venture Center. A senior scientist, Dr. Subhash Chavan from Organic Chemistry Division, CSIR NCL. Our sincere and heartfelt thanks to all the advisors who guided us and gave critical inputs for the entire matchmaker process and beyond. Today we are going to witness 7 pitches, 15 minutes each. The format would be each tech pitch would be for around 5 to 6 minutes. Then the scientist would speak about salient features of the technology for 2 to 3 minutes, and then the floor would be open for question and answer for rest of the 7 to 8 minutes. The intended outcome is to get expression of interest from industry people who are in the audience today. This is a free, non-binding expression of interest form which the seekers can fill. Our team plans to put link to this expression of interest form in the chat box while the technologies are being pitched. That is all about the matchmakers and the showcases that TechX team runs. I hope you enjoy all the technology pitches curated by our team. Thank you.
With that brief introduction, I would now invite Dr. Premnath to kindly set the stage and say a few words about the matchmaker. Thank you.
Well, good evening, everybody. Thank you for joining in. Very happy to run this particular showcase, especially in the pharma domain. It's very close to us. The Venture Center is hosted by the National Chemical Laboratory, Pune, and it's an independent incubator that is supported by a variety of different government organizations, including the DST, BIRAC, and various other departments for the kind of various different innovations in different fields as well. But that said, the pharma industry is especially important given its close association with the National Chemical Laboratory, and we are happy to welcome you here to this event as well. The idea here is to help create what I call useful collisions between academia and research labs and inventors on one side, and people who want to take it ahead and commercialize it on the other side as well. And in this effort, as Pragnya has mentioned, and which I'll reiterate, that this is entirely a pro bono effort from our side. It is meant to support some of these both parties, including academia, R&D, and industry. I'm glad you're able to participate. At the same time, this is also an effort where we are trying to present given academic achievement, which might be an invention which is useful in a format which is useful and understandable to in a language which industry is comfortable with. So I hope that is useful for all the concerned parties as well. The end point is that we'll try and try to take these connections we forged today and direct them to the labs and the industries and take it ahead. And that will be something which I hope results in some outcomes at the end of the day, which where there are industry academia collaborations. I want to thank all the advisors. We have realized over the years that such uh, showcases are not possible just sitting in one corner of the country, but it's actually people who understand the industry and who understand what industry needs, and on the other side also understand academic inventions, who can actually provide valuable insights into what might be of interest, why it might be of interest, and for whom it might be of interest. So that's what this process of selecting and curating and presenting has involved. So thank you very much, and thanks to also all of you for participating. Thanks to all the inventors who have generously provided their time, to our team members who have helped curate the pitches as well. So the format, as you will hear, would be first a short presentation on the merits and the benefits of the technology, and then followed by a short uh, commentary by the inventors on the uniqueness of their technologies, followed by question and answers, which will be led initially. We'll request our advisors to add a few comments and then open the floor to everybody. This will be fast paced. It's about 15 minutes. So each technology. So please bear with us and use the chat box to post your questions. I'll help facilitate this with our team members in making sure that most of the questions get answered. Thank you very much, and I look forward to this showcase. Over to you, Pragna.
Thank you, Dr. Premnath. We would now start with our technology pitches. The first one is molecule nintedanib, and this is from CDRI Lucknow, Dr. Panda. I would request my colleagues to kindly share the pitch, and then we can have 2-3 minutes session with the scientist where he can mention the salient features of his technology, followed by question and answer.
Good afternoon. The technology that I will be talking about today is a process for preparation of nintedanib. The lead inventor is Dr. Gautam Panda, and the organization that he belongs to is CSI or CDRI. Nintedanib, as we all know, is a small molecule TKI which is orally administered as a capsule. It is approved by US FDA for the treatment of idiopathic pulmonary fibrosis. EMA and PDMA have approved it for the treatment of non-small cell lung cancer. Currently, the cost of nintedanib in India is almost 5,000 to 6,000 rupees per day, which is unaffordable to most people, and the price of the generic version is around 85 to 90 rupees per 150 milligram tablet. What are the market opportunities? The market size for nintedanib was valued at USD 1.5 billion in 2023. The innovator industry is Boehringer, and the basic product patent has expired in 2023, and the secondary patents have exclusivity till 2029. The licensed manufacturers in India are some of them are Cipla, Lupin, Zydus, Cadila, etc., Glenmark, etc., and generics have been produced by Glenmark Pharma, Cipla Limited, etc. The existing process which is used has some drawbacks, like the total number of steps is more. There are 9 steps. Acetic acid, DMF and acetic anhydride have been used. The intermediate steps are difficult for synthesizing at a large scale. High temperature has been used. Hydrogenation is required for more than one type, and the yield of the reaction is not very high, in addition to harsh reaction conditions being used. There are various advancements and improvements to the process, which is a new improved process by CDRI, where the number of steps is lower, 4, and it is using more environment friendly reagents along with easy access to the oxoindole skeleton, and it is a more robust and scalable method with instead of 9 to 10 steps there are 2 to 4 steps, and using the domino reaction and less number of chemical reagents with the acceptable yield. So the estimated cost of raw material is USD 1919 per kg of the product yield. The current status is it is at a TRL level of 4 demonstrated at a 50 gram scale. The Indian patent has been filed with a publication date of August 23 and a priority date of February 22. They also have publications around this technology. What is the value proposition? There is a higher yield of 35 to 51% versus 35 to 40%. The number of steps is lower, 2 to 4 versus 9 to 10. Milder reaction conditions, eco-friendly solvents. No column chromatography is needed, so the OPEX is lower. A higher purity of 98%. Along with the residual palladium to 10.9, and the estimated raw material cost is comparable at $1,998 per kg, and this is included in the PLI 2.0 scheme. Who should be interested? Pharmaceutical API manufacturers who wish to leverage the PLI scheme, and companies who already manufacture in nintedanib and want a better process. Why they would want so is to make the because it is a more cost effective, simpler and scalable reaction with fewer steps required. The lead inventor is Dr. Gautam Panda, who comes with 22 years of biomedical research experience. He has worked on cost effective synthesis of blockbuster drugs. He has more than 132 publications to his credit and is the inventor of 10 patents. His industry collaborations include those with Dr. Reddy's, Natco, Reliance, etc. CDRI is a premium institute under the Department of Science, Scientific and Industrial Research. They have more than 13 new drugs which have been discovered and developed, and several success stories as you can see in the slide. They have various industry collaborations. As you can see, there are various big names in the slide for their industry collaborations. So the next step is it is possible to scale up this technology with support from industry partners from a pilot scale to a continuous flow if required. In partnership with other CSI labs, they are seeking licensees for technology and IP on as is where is basis, and joint development and sponsored R&D partners or scaling up the technology and advancing it. For more details, you can contact me at the connects which are written in the slide. Thank you.
Requesting Dr. Panda to kindly unmute himself and talk about the salient features of the technology. Can you see the slide on the screen, sir? Dr. Panda?
Yes, I can see.
Yeah. Please go ahead.
Yeah, so thank you very much, most particularly Venture Center, for giving us an opportunity to tell something about our lab scale technologies of nintedanib with your industry persons. If you see the nintedanib, that a known process route, as has been told by earlier person, that is nearly 9 to 10 steps, and we have done two approaches. First approach consists of nearly 4 steps, and second approach we have come down again to just 2 steps. And currently these approaches we could yield the nintedanib from 50 to 70 gram scale. This has been demonstrated, and we have checked the residual palladium impurity, that is just 10 ppm at the current moment, which is acceptable at the industry level. And if you see the overall yield in comparison to the known method and with the CDRI method, it has drastically come down from known to nearly 35 to 40%, and we got the yield nearly some 51% at the higher side. And This reaction, this scalar lab scale technology does not require any column chromatography, and it has been shown that very critical multi-component reaction can be used so effectively to gather the skeleton of oxindole, which is a part of the nintedani. So we are looking for the industry partners, and we want to give this technology as is and where is at level. So that's all from my side.
Yeah, thank you, Dr. Panda. I think with that, we will move on to comments from any of our advisors. If Pragna, did you are you allowing
Yeah.
opening of the
Yes. Now we can request advisors to comment, and in the meanwhile, if we get some questions in the chat box, we can take up those after that.
yeah. Please feel free to type your questions in the chat box. Dr. Singh, do you want to go first?
No, I think Premnath, this is fine. I don't have any questions. You can ask, other.
And also, besides questions, if you want to comment on any specific step here, that also you can do.
This is all fine. This has been discussed on a separate platform at
Yeah.
CSIR. Okay, so all look okay.
Yeah. And also, what would be, Dr. Panda, what are the typical next steps that you expect would be necessary to take it to market?
So actually that we are expecting some of the questions and questions from the industry partners, and this technology whole details has been shared with Dr. Reddy's at Hyderabad. So they are currently evaluating it, and we at the same time we are also looking for other industries if they want to take these technologies at the present level and then again evaluate at their end. So that is what we are looking for, because this is anti-cancer drug. As you can fully understand, we are not supposed to scale up more than 100-gram scale at our laboratory level because we don't have that precautionary measures. Because it's anti-cancer molecules, so this requires a very specialized facilities. So basically, we are looking for the industries who can take up this technology at this level itself, and then basically evaluate there, and then again come back to us that what are the things has to be addressed.
Okay, so there is a question, but before that, I just wanted to ask if any of the other advisors want to comment anything, either Dr. Nair or Dr. Chauhan.
So I have no comments to make. We have already discussed this earlier.
Yeah. So in this particular case, there's a question about somebody asking that would it be possible to give a plant scale batch demonstration for this product if we collaborate with each other? This is a question from Rajesh Chaudhary. Let me take this question a little bit as well. So CDRI, as Dr. Panda mentioned, takes it to a certain level. But of course, within CSIR, there are capabilities for advancing it. So there are industry partners who want to collaborate. Then CDRI, along with one of the other labs, which is focused on process development, can collaborate to try and take this ahead to the next level. But I think industry interest is the first thing that they would be looking for. Do you want to add anything, Dr. Panda?
Yes, actually, we are. This has been discussed with our business development team, and this Dr. Rajesh Chaudhary, he is most welcome, and he can write a mail to me, and then we can separately discuss and do with business development group, and we'll get back to them. As there has been pilot plant scale a demonstration at CDRA level, it's not possible. This has to be done with other CSR laboratories, so that discussion we are always open.
Yeah, I think there's a request for recordings after the meeting, which I think will be available. Will be available definitely. And this is typically done soon after the meeting as well. It will also be mailed to the people who have registered for the event as well. right? Okay. So in this particular case, I just thought I will, Dr. Singh, did you want to comment, say, for example, on the cost that has been estimated? Is that a fairly competitive price?
So, Dr. Panda, what's I can't see. This is US dollar. How much is 1919 per? I could not.
Kg. Yeah. 1919 per kg.
This is this is the raw material cost.
Yeah.
Looking very competitive. Looking at the market price. Okay.
Yeah.
Okay.
So price looks okay. The only thing is the final quality. Is it the qualitative or quantitative? Of course, Dr. Panda, I could not ask. Maybe many industry partners would be looking that. And that's the one. And all the impurities. The final quality along with the likely impurities related substances. That's the one. And second, of course, CSIR labs, they are, basically powerhouse for, PUC and other. So gentlemen who asked for the demonstration at plant, I fully agree with Professor Panda that that is not possible, but that is possible through industry partners, But they can definitely demonstrate at a scale they have developed this process, maybe 5 gram, 10 gram, or 50 gram. I'm sure Dr. Panda would agree to that.
Yes.
Yeah.
So
Dr. Panda,
Dr.
do you
Singh, I
want
think
do you
we
want
have
to
discussed
comment
with
on
you
the
also.
do you want to comment on the question about quality as well, which is that what do you have some numbers on which you are mentioning that the quality is high or
Yes,
some measurements?
yeah, actually, a final product at 50 gram scale, we get more than 98% purity, which is required by the industry. And we also check the residual palladium impurities, so that is within 10 ppm, so that is also within industry range.
Yeah. There's also a question about from Dr. Sudhir Sawant. How do you get intermediate A, which is you can see on the screen? Do you want to comment on that, please?
Intermediate, can you please go back show the slides?
Yeah, you can see it on the screen right now.
Yeah, okay, yeah.
That's the one which is on the left side here on the screen.
Yeah, no, actually, that intermediate A has to be obtained. So intermediate A and also intermediate 8 is same.
Yeah, so that is synthesized by you through these various steps is what I think you wanted to say, right? So can you just go back one slide, Pragnya, just to show the scheme? Yeah, so sorry,
No.
I think, sorry, go back to the next slide. I think, Dr. Panda, the question is coming to number 8. Do you want to describe the reaction which reaches number 8?
Yes.
Yeah.
So that also reaction is also shown here. So this is basically acid-amine coupling. And so that can be done very high scale. And after getting that your next step, which is circled by, which is basically circled, that is a kind of the crucial reaction which is required for the intermediate. So, so as for the question, the intermediate 8 and intermediate A is the same, and intermediate A has to be synthesized from starting material 6 and 7, both which are all commercially available.
Yeah. Dr. Kishore More has a question. Is the final API, is it a ICH grade material?
Final API must be ICH grade. Yeah, because we checked only the purity level.
Yeah.
And what is ICH grade and all, yeah, this final material definitely this is ICH grade only, because we have checked the 98% purity at 50 gram scale.
Okay, so I think if there are any other questions, please put it on the chat box. We'll be happy to take it. In the meantime, is Pragya, is Dr. Nair also attending?
No, I think he's traveling. So
Okay.
he was not sure whether he would be able to attend or not. Yeah.
Okay, good. Okay.
Yeah.
There's also ICH means all impurities are less than 0.10%. That's what Dr. Sudhir Sawant has looked to.
Yeah, yeah, because see, with this particular lab scales technology, we still did not do the impurity profiling. After getting the final compound, we have checked that at present residual palladium impurity, and we also checked also one more impurity just that we have characterized. A detailed impurity profiling, this is not yet done.
Yeah. So I think if there is so according to Dr. Sudhir Sawant, just 98% purity
Yes,
is
sir, I
not
fully
sufficient.
understand.
Yeah. So at the industrial scale, yes, you will have to do all of those things. But I think at this time, this is essentially meant to be an alternate process for this particular molecule, right? Prashant Hebar has a question, which is the target receptor for the final molecule?
Actually, this is a broad spectrum kinase inhibitor, so which is approved actually by European Medicine Agency for lung cancer and for US FDA for IPF, means idiopathic pulmonary fibrosis.
Yeah.
And this is a triple angiogenesis kinase inhibitor.
Yeah, I think there's one other question which I'll take up. Do you provide process? In the meantime, Pragna, you can just go back to the first slide, which had the description of the molecule.
Yeah,
Okay.
is there?
Yeah, this one.
Yeah,
Okay.
yeah.
Do you provide process development report also suitable for the purpose of DMF filing? I think, Dr. Singh, is this something which the industry will do? Partners eventually end up doing? What I
Yeah,
personally don't
yeah,
know the
because
details of what all goes into a DMF filing.
I agree with you because, see, the primary purpose of CSIR labs is, the knowledge generation, new chemistry, or, very cost-effective chemistry. And then, is a kind of proof of concept. And then, at a gram, they have developed the technology. There are many regulatory angle and, IP angle and other thing. Perhaps, that industry will be able to do it with the collaboration with the CSIR lab.
So typically what happens is that once the process is demonstrated at a certain scale, there is a process of scale up and hitting all the requirements, additional requirements that industry has. This could be done either by another lab or a company as well. Depends upon if there's an intermediary company which is scaling it up and then providing it to larger companies as well. So this is not meant to be a ready to deploy technology at scale on day one. It is meant to be advanced in that direction. That is the purpose here. Dr. Panda, there's one other question. This is from, I think, Swapnil Zadbuke, which says, thionyl chloride has environmental hazard. Is there any other way to overcome it? Can you go back to the reaction scheme, Pragna? Yeah, please go ahead.
I think we have to go to the next step.
Next slide, yeah.
Yeah. So thionyl chloride, we fully understand that it has environmental hazards, and to replace that, we have tried with other coupling agents. So they were not as fruitful as SOCl2. So this one we could not replace. We could not replace SOCl2 with other coupling agents, and that also will be giving a better yield. So currently, this is under investigation.
Okay, I think with that, we probably have run out of time. Pragna,
Yeah.
can we move on to the next
We
one,
can
please?
move on to the next one. We can have few
Please
more
feel free
questions
to
towards
send.
the end.
Yeah, you can send your questions. I will just mention which technique it is, which process it is for. We'll try and take it up later. Yeah.
Next, we have para-aminophenol from Dr. Sanjay Kambre requesting my colleagues to kindly share the next technology pitch.
In the meantime, we have put a form which you can fill up if you're interested in discussing further. We do understand that many of the academic technologies will be at not all, but most of them would be at a smaller scale and need to be scaled up. But I think the key here is innovative process technologies which are competitive is what we're trying to present here as well, and then can be taken further ahead by industry. Right? The de-risking process is typically best done by the industry. Yeah. Continue, please.
Today I am going to present on behalf of Dr. Sanjay Kamre from CSIR NCL a cost-effective and green process for the synthesis of para-aminophenol from para-chloronitrobenzene. Para-aminophenol is key intermediate in the production of paracetamol. Production of p-aminophenol is included in PLI scheme of Government of India. The price at which we get PAP in India from China is $1.5 to $2.5 per kg. The global para-aminophenol market size was valued at USD 620 million in 2023. By end user segmentation, pharma industries have the majority market share. The innovator product patent is from L. DuPont de Nemours and Company. Priority date was back in 1952. Hence, there are many generic players now manufacturing and selling PAP in the market. The existing commercial process is a two-step process where the starting material is para-chloronitrobenzene. Here is the reaction scheme which you can see on the screen. The NCL process is different as it uses direct hydrogenation of sodium nitrophenolate, a two-step reaction where the step 1 is synthesis of sodium nitrophenolate from PNCB and step 2 is synthesis of para-aminophenol from sodium nitrophenolate. The advantages of this process is that PAP productivity has been increased more than 20 times compared to the conventional process owing to highly soluble sodium nitrophenolate that is formed in water. Effluent generation has been reduced by approximately 50% compared to the commercial process. A non-noble metal catalyst has been developed which remains effective for multiple recycles. Wastewater generation has been minimized via recycle and reuse of alkaline water in the process, therefore no waste generation. The product obtained is a stable light brown color. Synthesized product PAP has purity of 98 to 99% with yield of 85%. The estimated raw material cost is around $2 per kg for the NCL process. Which industry and who should be interested in licensing the technology? Paracetamol producing drug companies, because they have heavy reliance on China-based companies with highly fluctuating costs. Therefore, most of the pharma companies wish to manufacture PAP in-house. Petrochemical industries, they are looking to diversify into chemical and pharmaceutical manufacturing. As part of this strategy, they have identified certain molecules. One of them is PAP. Current status of the technology, it has been demonstrated at TRL 4.5 kg scale. The innovation is patented with a priority date of 2023 and coverage in India. Value proposition of the NCL process, they are using non-noble metal catalyst which is recyclable multiple times. More product is formed in reactor of same volume, resulting in better productivity per reactor volume. Yield is around 90%, purity 98 to 99%, which can be improved further. Water is used as solvent and can be recycled, and the estimated raw material cost is comparable to the landed cost, that is $2 per kg. The lead scientist, Dr. Sanjay Kamre, he is Senior Principal Scientist, CEPD Division in NCL Pune. Some areas of his expertise are process intensification, depolymerization of plastics, conversion of biomass into valuable chemicals. He is recipient of various awards and honors, to name a few. NCL RF Sipla Hamid Award, he has won consecutively for 4 years. CSIR Young Scientist Award, he is Life Fellow of Indian Chemical Society. The process development group at CSIR NCL is having well-equipped laboratory facilities. They have strong collaborations with academia, industry. They have significant number of patents and technology transfers to the industry. They have won Best Commercialized Technology Award for the continuous process for production of paracetamol during the year 23-24. On the screen, you can see certain photographs of infrastructure and analytical facilities that are available with the institute. They have track record of industry collaborations. This is a snapshot of industries whom they have worked with in the past. The scientist has developed lab scale process for preparation of PAP. He has also developed downstream processing methodology for purification of PAP. Impurity profiling of PAP has also been established. Now he is looking forward for industrial partners who are interested in know-how transfer or sponsoring further technology advancement and scale up or licensing the technology as is where is basis. If you have any further questions, you can contact me. My coordinates are given on the screen. Thank you.
Requesting Dr. Kamre to kindly unmute himself and speak about the salient features of his technology.
Will you be putting up some slides? Yeah.
Yeah, I'm just putting up.
Hello.
Yeah.
Good afternoon. Thank you, Dr. Premnath and Pradnya, giving me opportunity to present our work on this platform. So already we have covered most of the advantage of the process what we are developing in NCL. Basically, as you have mentioned, para-aminophenol is a key raw material for production of paracetamol. In India, requirement of PAP is around, you can say that 40,000 per annum, and we are importing from China. So many industry they are looking towards sustainable process for the production of PAP. There are two routes basically, nitrobenzene route and another route is PNCB route. So in NCL we are interested in PNCB route. There are multiple reasons because in India PNCB is available and basically nitrobenzene route already has been investigated in NCL and there are some limitations. So I'm not going to the detail, but what I'm saying that NCL recently we have developed a sustainable eco-friendly process for the production of PAP. Basically, we have got here yield is more than 85 to 90%, and impurity profiling also we have done. And basically, here wastewater generation we have reduced, and we have whatever the wastewater we have generated, we have recycled and used multiple times. The catalyst, whatever we are using in this process, is non-noble metal catalyst, and which is also recycled, reused multiple cycle. And we have done the cost calculation, and we found that our process is much efficient and cost effective as compared to a conventional route. Now we are in the process to develop continuous process for the production of PP. Basically, there are two steps, and first step presently we are working to make a continuous, and as well as second we are hydrogenation also we want to do in continuous mode of operation. So in next 6 months, I think we are getting one reactor where we want try this process in continuous mode. And already Pradnya has mentioned that Indian patent has been filed for the same. Thank you.
Yeah. Thank you, Dr. Kamble. I'll just go to the questions, but before that, I just wanted to request you to discuss a little bit also about the fact that in the same reactor volume, you're getting considerably higher productivity, right? Do you want to comment on that and give some numbers around it, please?
Yeah, basically, we are not using, in this process, we are using sodium nitrophenolate. And sodium nitrophenolate has higher solubility in water as compared to PAP. PAP solubility is only 5%, but if you have sodium salt of nitrophenolate, then solubility may be 25%. So automatically you can enhance your productivity. Because in conventional process, what they are doing, PNCB, they are converting into reacting with the NaOH and they are getting sodium nitrophenolate and they are doing acidification. Then you will get PAP and further they are doing hydrogenation. So we are not followed the same route. What we are doing, we are converting PNCB into sodium nitrophenolate salt and directly we are going for hydrogenation. And that's why we are getting more productivity in the same area.
Yeah, and why is that important?
Because otherwise suppose if you want to produce 1 ton of PAP, then size of the reactor is very big, then your capex and operation cost will be very high.
There's a question I think before that let me just ask the other advisors if Dr. Singh wants to comment anything about the technology, including highlighting any specific points in the technology, then you can, and I'll also request Dr. Chavan to do that as well.
Yeah. So thank you, Dr. Kamble, and this is a good work, But looking at the Indian reliance on Chinese import, I understand that you said chloronitro benzene. You're starting from chloronitro, correct? Now
PNCB,
the
para-nitrochlorobenzene.
the requirement of India and the kind of PLI scheme, we are producing only 40% of the chloronitrous. Still 60% is coming from China. Okay, so this in order to be successful, we have to start from basic chemicals, available in India, particularly from benzene. If we can start, from benzene to chlorobenzene, that would be great. That's the industry's, pulse. I talked to many people, and they said, if we really want to be successful like China, okay, we have to start from benzene and then benzene to chlorobenzene and subsequently to your this thing. Now, second, most important, China, of course, three companies, out of three companies, two companies are working on the continuous process. And I have a first-hand information, their raw material cost, RMC, okay, is close to 150 to 160. And most of the Indian, pharmaceutical partners, they are looking for a raw material cost is close to 150 to 160 rupees. I understand, you are saying somewhere, close to $2. I heard someone saying $2 is the RMC. So your comments on that.
Dr. Singh, I have discussed with couple of industry. PAP price is fluctuation. Maybe sometime it is 160 to 200 rupees. Definitely our price is less than that, but we have considered whatever may be PNCB cost, then whatever NaOH cost. So there is a fluctuation, but definitely our process, there will be profit around 20 to 25%. That is sure. Because we are using utilization will be less. And second thing, you want start from benzene. Already a couple of industry like Aarti industry, they have already developed process for benzene to PNCB. So raw material is easily available for PAP. So
Yeah.
there is a request from industry. You should start with raw material PNCB instead of going to benzene, because already this is established one, benzene to PNCB.
Okay.
Yeah.
Okay. Go ahead, Premnath.
Okay,
I
good.
don't have any
Yeah,
comment.
no, but those are good comments, Dr. Singh. We'll see how we can do that. Let me move to Dr. Chavan as well. Dr. Chavan, you wanted to comment anything before we move to the questions?
Oh, I only wanted to comment that this is a development where the salt itself is hydrogenated and the solubility is increased in water. So that is a very plus point of this process. I would like to ask how many times it can be recycled, although Dr. Kamble said multiple times. I would like to know that.
In NC, yeah, in NCL we are recycle at least 10 times. But
Okay.
still again you can recycle. So there is no any issue. At least we have done 10 cycle in NCL.
Is it a
I suppose, yeah. Sorry, please go ahead, please.
is it a heterogeneous catalyst?
Yeah, heterogeneous catalyst.
Okay.
I think that covers Dr. Veera Reddy's question of the metal catalyst turnover ratio, right? Or do you want to add something more, Dr. Kamble?
That detail I will share you later on. If you want, we have that information. Detailed discussion. Once you agree, then we can share all details. Turnover number, impurity profiling, everything is available. Once that agreement is signed, we can discuss all the detail.
Yeah. So Dr. Kishore Mori has a question. Which catalyst is used for NO2 to amine? Can I just, yeah, this
Basically,
catalyst.
basically we use non-noble metal catalyst.
And this is subject matter of your patent, right?
Yeah.
Yeah. So this is currently has been filed. It is in the, I mean, that's as is listed over there as well. Dr. Sudhir Sawant has a question for PAP. Which could be the best process, traditional hydrogenation or flow chemistry?
we are planning continuous process. That because flow continuous process has many advantages. Raw material consumption per kg product will be less, and then quality of the product will be consistent. So we are planning go for continuous process.
And how far are we from getting to a stage where you'll have a continuous process?
It will take time because initially impurity profiling and all these things have taken time. But now I think within 6 to 8 months we'll get the reactor, and after that I think this year we are planning to establish continuous process within 6 to 8 months.
Okay, great. Thank you. In the meantime, while that is happening, I think I'll request Dr. Kavita to Parik to put the form. Please feel free to put in your. So as I mentioned, this particular form is a non-binding expression of interest. It's a very brief form. It just has a few things. It's just to help us make those connect post the meeting. There's no other purpose for it beyond just helping set up some discussion meetings and so on. So that I think I'll request people to take it up. If anybody wants to talk about this, they can just fill up this form as well. So are there any other questions if I should take them up?
In
Then
the
I would
interest of
otherwise,
time, can we just take it at the end?
sure.
Yeah,
Yes,
yeah,
please go
yeah.
ahead, Pragya. Yeah, you
Okay.
keep track of time. Yeah.
Yeah. Thanks, Dr. Kamre. Thank you
Thank
so
you, Kamle.
much. Please be on the call. We may have questions at the end.
Okay, thank you.
Yeah, thanks.
Thank you.
The next one in the list is molecule from sacubitril, that is Dr. Ravinder Kontham, NCL. Requesting my colleague to kindly share.
Heart failure, a chronic and debilitating condition, affects millions worldwide, posing a significant burden on healthcare systems. In the ongoing quest for effective treatments, the combination of sacubitril and valsartan has emerged as a crucial therapeutic intervention for adults and even children with symptomatic heart failure, helping to reduce the risk of death and hospitalization. Valsartan, an angiotensin II receptor blocker, plays a vital role in this combination therapy. It blocks a substance in the body that causes blood vessels to tighten, thereby relaxing them. This action leads to lower blood pressure and an increased supply of blood and oxygen to the heart. Sacubitril is a neprilysin inhibitor, and its synthesis, particularly the production of its intermediates, is a complex and critical process in the pharmaceutical industry. The market for sacubitril-containing drugs is substantial and growing. In 2023, the global sacubitril/valsartan sodium market size was estimated to be a significant $2.9 billion. This growth is primarily driven by the increasing prevalence of heart failure and the growing demand for effective treatments to manage this chronic condition. Several major players in India are actively involved in this market, including Novartis, Lupin, Dr. Reddy's, Cipla, JB Chemicals, and Pharmaceuticals. Despite the market potential, the production of sacubitril and its combination with valsartan faces certain challenges. Notably, Novartis holds patents related to using sacubitril and valsartan for chronic heart failure. Additionally, Merck has patents concerning the production of sacubitril itself or its prodrugs, as highlighted by the patents shown on the slide. These existing patents can create complexities for other manufacturers looking to enter this space using various production processes. Recognizing these challenges and the importance of sacubitril in treating heart failure, Dr. Ravinder Kontham, the lead inventor from CSIR NCL, has developed a novel process for the synthesis of sacubitril intermediates as shown in this scheme. This innovative approach offers a promising alternative to existing methods. The total RMC for the final product sacubitril is $1,103, and the key compound, compound 10, has an RMC of $503. Dr. Kontham's process presents several compelling advantages. Traditional methods, such as those using a Grignard's reaction, can suffer from unfavorable stoichiometries of reagents, making the resulting product expensive to produce. Dr. Contham's modified stoichiometries and reduced catalyst quantities make this new method particularly attractive. The process utilizes a chiral starting material that is easy to work with and a standard, readily available building block. This new methodology also leads to reduced purification steps and consequently lower costs. Furthermore, it requires reduced quantities of catalyst and other materials that are safe to use in the lab, further contributing to cost reduction. The technology developed by Dr. Kontham is currently at a technology readiness level of 4, with lab level results successfully demonstrated. The team has produced intermediates at a 10 gram scale and the final sacubitril product at a 1 gram scale. Further evidence of the innovation's potential is its inclusion in the production-linked incentive scheme from the Government of India, signifying its importance for domestic manufacturing. A patent has been granted, and research on this innovative synthesis has also been published in peer-reviewed journals. This technology presents a significant value proposition for active pharmaceutical ingredient manufacturers. Currently, sacubitril and its intermediates are imported from China at high cost. Dr. Kontham's process offers a pathway to cost-effective indigenous production, reducing reliance on imports and potentially improving supply chain resilience. The research team at CSIR-NCL has laid the foundational scientific work, demonstrated lab-scale feasibility, and established proof of concept. They are now seeking industrial partners interested in technology licensing, sponsoring further technology advancement and scale-up, or raising third-party funds for a collaborative project. They are also open to industries interested in leveraging their scientists' capabilities as experts or consultants. The next phase involves close collaboration with industry partners to define techno-commercial specifications, optimize the process to meet industry requirements, and undertake scale-up, further optimization, testing, and certifications. CSIR-NCL and Dr. Kontham's lab boast state-of-the-art laboratory facilities and a team of eager scientists ready for scale-up. Dr. Kontham's innovative process for sacubitril intermediates represents a significant advancement in pharmaceutical process innovation. For more information or to explore partnership opportunities, please contact the name and number mentioned on the slide. Thank you.
Requesting Dr. Kontham to kindly unmute himself. And you are on mute, Dr. Kontham. Can you speak again?
Can you hear me now?
Yeah. Please go ahead. Please go ahead.
Yes. So thank you, Dr. Premnath and Prajna, for giving me this opportunity to present our work here, this very important platform. So first of all, I would like to cite that Dr. Chavan, who is in the expert committee, also worked on this project. He is also one of the innovators in the patent what we filed. So unlike other molecules what already presented in today's meeting, this molecule is a little complex molecule, sacubitril. It has two chiral centers at C2 and C4. One is amine chiral center, another one is methyl chiral center. So in general, if you see all innovators processes, so they use asymmetric hydrogenation as a key step to install the amine chiral center. So which needs iridium, ruthenium, rhodium like highly expensive noble metal catalysts and high hydrogen pressures. So one of the basic process route for this sacubitril reported by Hook et al. From what is that? So they used a chiral pool building block. Chiral pool building blocks are natural products which already having existing chiral center. If you see the starting metal one, pyrrolutamic acid, it is a chiral molecule. It is in chiral amine center. And this is already fixed by nature. So this is a very cheap starting material, pyrrolutamic acid. So in the innovators process, if you see the intermediate 3, so in our route we have N-benzyl group. Suppose if you see innovators route, there is a BOG group is there. So the BOG group is very sensitive towards the coupling reaction to make yite. Okay? So we tried first we optimized our reaction conditions and innovators process. And we found that there are many difficulties in innovators process. If you have a bog group, so generally the intermediate 7 is there in the box that should be converted into Grignard reagent and should be treated with a tosylate or iodo compounds, 5 or 6. So instead of benzyl, if you have a bog group, that is very sensitive material and coupling was very difficult. So another thing is the intermediate 7, we need to convert to the Grignard reagent. If you see the innovators process, they used this biphenyl bromide intermediate 7 and copper cyanide and magnesium. So this complete cocktail they used in 4 equivalents, 4 stoichiometrics, 4 times. You see 5 or 6, 4 times you need 7 Grignard reagent prepared from 7. So, and even that reaction is not selective. If you have an N bog group, that leads to many impurities and opening up the ring system, the 5-member lactone ring. So then we focused on exploring various protecting groups on the 5-member lactone intermediate 5 or 6. So after several investigations, we found that the benzyl group or paramethoxybenzyl also very good protecting groups that can tolerate the harsh reaction conditions where a Grignard reagents can be used. Even we tried with this intermediate 5 and 6, the low Grignard reaction conditions, and as well as we developed our own Grignard type reaction conditions. So why? Because the this particular Grignard reagent which is used in this process needs diethyl ether as a solvent, so which is having very low flash point, and we need to reflux it to make a Grignard reagent. So once the Grignard generates, okay, that is insoluble in diethyl ether. Again, we need to change or add any other polar solvent to the Grignard reagent. We need to add it. So these are the very big difficulties in the innovators. So we developed a process. Suppose if you take intermediate biphenyl bromide 7 and a catalytic amount of copper iodide, DPPM is the ligand, okay, and lithium oxide is the base, and magnesium. So there is no need to eat anything. Just we can take intermediate 5 or 6 and we can mix with 7. We can add this all copper iodide, DPPM, lithium oxide, and magnesium in a one pot. So instead it generates the Grignard and it couples with intermediate 5 or 6 and generates intermediate A. Yeah, so once intermediate A is in hand, so intermediate intermediate 8 having one amine chiral center. So we need to fix another one chiral center, the methyl group at alpha to carbonyl. So this is through substrate controlled stereo selection. We tried several bases for this. Finally, we found a very good base like LDA, KHMDS, lithium HMDS, and we able to do methylation dash stereo selectively. Okay, and we obtained intermediate 9. So the 9 we converted to the cyclic lactam 10. This 10 intermediate is available from China. So really we don't have exact figures, the import cost of intermediate 10. So our manufacturing cost, RMC cost is not manufacturing cost, sorry. RMC costing is 502.9 US dollars per kg. Yeah. So this is a little multi-step process. So unlike earlier processes, we need to do 8 steps to access intermediate 10. So if any industry is interested, we are happy to collaborate. And even so, based on RMC calculations and industry economics, we can still further work on it to improve the existing process. Okay, we can do some alterations based on industry requirements. Thank you very much, Pragya.
Thanks for your comments on the technology, Dr. Kontham. I would now request Dr. Girit Singh and Dr. Chavan to give their comments, overall views about the technology. Dr. Girit Singh.
Yeah, no, it looks good, Dr. Chauhan was, as I heard, Dr. Kontham, he was one of the inventor. One question, on behalf of the industry, Dr. Kontham, have you benchmarked for quality your product with the product available in US or in India, particularly through the Novartis? I'm asking about the quality benchmark of this sacubitril, the sample you prepared in the lab.
Yes, sir. So we prepared sacubitril with 99.5% purity. And we compared the data, some advanced intermediate, a bulk intermediate also we prepared after 10. So if you treat with hydrochloric acid, the lactam gets opened, and then we protected that preamble with POC. So that is also commercially available. So from that intermediate, even we synthesize sacubitril in more than 10 gram scale. So, and we compared the data with the commercial sample. So it is 99.54% we got it. And in other words, the sacubitril we did multi-gram scale, even Dr. Rajesh Gunade, this is not as part of this invention disclosure. So HCL developed very novel polymorphs also for formulation purposes. So we match the data with the reported.
Yeah, I appreciate that. This is the one product, where multiple filers, particularly for US market, I understand more than 16. Of course, our industry partners can add them. And this is very important product for country. Okay. Now, what matters is the cost. Okay. So we will tell you, very, very briefly, the sacubitril valsartan, that's a co-crystal, you understand. Okay.
Yes.
That price is close to, $250 to $300 range. And the price component of sacubitril in that, if this turns out to be about $250 to $300 a kg, okay? Let's not discuss about the IP because there is a IP, definitely. Whether it's an infringing or non-infringing, patent getting expired, shortly. So where are we in terms of costing, because, most of the industry players, they are looking for a very cost-effective process of sacubitril.
Yes. So in this process, lithium HMDS is the really cost contributor. So another important cost contributor is sodium iodide. When we make iodide, intermediate 5 to 6, sodium iodide also one of the cost contributors. So if we are able to avoid these two reagents, really we can down the cost, 50% reduction we can do.
Okay. That's all, Pratnya, from my side.
Thank you, Dr. Singh. Dr. Chavan, you wish to add here? Question
I have for Dr. Kontam is,
Sir.
what is the efficiency or the yield when you do the coupling with tosylate versus the iodo?
Iodide is better, sir.
Okay, how much better?
So we are getting 75% with iodo.
Okay.
So in case of 5, we are getting 65%.
Okay, fine.
Yes, sir.
Thanks.
There are some questions in the chat box. You can take those, right?
Yeah.
There's a question which refers to a specific patent. I think we may not be able to discuss this right away unless Dr. Kontam recognizes this specific US 1016 patent, right?
Yes.
So I think Dr. Patak, I have sent it on. Yeah. So I think I have the patent reference. You can go back and check it separately. May not be able to do it immediately, but
Yes.
maybe you can address other questions. Which salt of sacubitril is synthesized, and what would be the cost?
Yeah, the
Pragna,
final-
did we have numbers, RMC numbers here as well? Yeah, please.
Yeah,
go
we
ahead.
have those.
Yeah, Dr. Kontam, you want to talk about which salt?
I think it's HCl salt.
Sorry?
This is hydrochloric acid salt.
Yeah, and the RMC cost is listed here. Another question is about sacubitril being very prone to hydrolysis. What is the level of hydrolyzed impurity in sacubitril?
Yeah, actually, finally, we are doing the esterification. After opening with succinic anhydride. So first we are doing the esterification, then we are doing succinic anhydride. We could not find any hydrolysis product in the final step.
Okay. So I've this particular patent mentioned by Dr. Patak. I'm just sending it to you on this. You can have a look and maybe we can take it up a little later if time permits. I've also sent it to Dr. Nitin Shukla. You can also have a quick look as well. Okay.
Sure.
Are there any other questions or we can move?
Yeah, we have one last question. Then we can move on to the next pitch. Mr. Sawant wishes to know the enantiomeric excess obtained from intermediate 8 to 9. If you have that number, otherwise.
Yeah, it is more than 99%.
more than 99%, 8 to 9. Okay.
Yes,
Yeah.
yes.
Okay. Thank you. I'm
You
just
can-
sending you that link. You can have a look in the meantime, and then we can comment on it later. Okay.
Sure, Prerna. Thank
Thank
you.
you. Please go ahead. Yeah.
Thanks.
Just watch your Zoom list.
Yeah. We'll move on to the next pitch. Thank you, Dr. Kontham. Please be on the call. We will have question and answers sessions in the end as well for the rest of the questions which were not asked here. Next, we have Santa Quinn from CDRI, Lucknow. I would request my colleagues to kindly share the pitch.
Good afternoon. The technology that I am to present now is an improved process for Sentaquin. The lead inventor is Dr. Ajay Kumar Srivastav from CSR CDRI. Sentaquin was invented in 1989 as a hypotensive agent by CDRI. It was further developed by US-based farmers and it got approved in 2020 under the brand name of Lifaquin for the management of hypovolemic shock. Dr. Reddy's currently has entered a licensing agreement with farmers to commercialize it in India. Every year, around 1.7 million Americans suffer from hypovolemic shock, of which 348,000 suffer from severe symptoms and are eligible for Sentaquin. The market size estimate is approximately $40 million per year in USA. And in India, there are estimated 2 million patients with a market cost of approximately $200 million. There are various clinical investigations going on for Sentaquin for other indications like septic shock, acute kidney injury, cardiac arrest, COVID with ARDS, etc., which makes it a very interesting drug for process development. And Pharmas is currently undertaking a lot of clinical trials for different indications. The currently used methods have certain limitations. The method by Dr. Gulati et al. Has a yield of 73%, where there is use of acid, high temperature and has a moderate yield with impurities. Another method which is reported by Hanpo et al. Shows limitations of using a catalyst, high temperature and expensive starting material. The CDRI method has advantages in terms of yield, which is greater than 90%. It is an aqueous process, no acid catalyst is required. It can take place at room temperature with no column purification required. It reduces impurity formation, it has a more greener synthesis process and is higher yielding. The current status of the technology, it is at TRL 4 with the lab level results shown and demonstrated at 50 gram scale. The Indian patent has been filed with the priority date of 2021 and they have publications around this technology. The value proposition is that it has a higher yield of greater than 90% versus the traditional methods which give a yield of 47 to 71%. It has a much lower estimated raw material cost of $224 per kg. There is higher purity at a lower purification cost and the green credentials in terms of it being used in an aqueous phase, operational at room temperature, having a better carbon efficiency and E factor, as you can see in this table which gives a comparison between the existing approaches and the approach by CDRI, which shows that it has better green credentials. Who should be interested? Pharma companies which are interested in making and selling Sentaquin approved indications because it is a cost-effective, high-yielding method to produce Sentaquin. The lead inventor is Dr. Ajay Kumar Srivastav. He holds a master's in chemistry from Delhi University and a doctorate in chemistry from Jadavpur University. He has postdoc experience in the field of chemistry done from Seoul National University, South Korea. He has published more than 54 research articles and supervised 10 PhD students. His group has transferred Umifevinor to Medizest for repurposing, and also he is working with Dr. Reddy's lab and with Arkin Private Limited on various sponsored projects. The organization CDRI is a premium institute under the Department of Scientific and Industrial Research, Government of India, They have various success stories, as you can see in the slide here, and with more than 13 new drugs which have been discovered and developed by them. They have several collaborations, as you can see from the slide here. There are various big industries involved in collaboration. The next step, the technology has been optimized at a multi-gram scale, and with complete characterization of API and related impurities. The citrate salt has also been prepared, and it is possible to scale up with support from industry partners and in partnership with other CSR labs. They are seeking licenses for the technology and IP on as in various basis, and joint development and sponsored R&D partners for scaling up and advancing the technology. For more information, you can connect with me at the information given in the slides. Thank you.
Requesting Dr. Srivastava to kindly unmute.
Pragna, there's
Yeah,
a little bit of an echo.
hello.
please
Yeah,
go
good
ahead.
evening, everyone. Am I audible?
Yeah.
You're audible.
Yeah, okay. Yeah, thank you so much, Venture Center, for giving me the opportunity and presenting our technology to this dais. And I think during the process, we kind of the previous discussions that we had with a lot of people from Venture Center, that was a great learning. And I think also it improvised us in understanding our technology in a better way. So this particular process that has been described by Dr. Kavita, so you can clearly see that the final coupling reaction between vinyl quinoline and the particular amine that happens in water, and it doesn't require any acid catalyst and high temperature. Because of that, actually the other two processes also we did in our lab, and we checked that these two processes that were developed by previous inventors, they actually in that two impurity formation occurs, and that impurity formation was completely minimized in our process because it happens in water and there is no alcohol used, and because alcohol also participate in the reaction and generates the impurity. So these are the features that have already been described. Apart from that, we also took up this particular API and then converted it to citrate salt that is actually used in the approved drug. And for that also we generated the API specification. And as per COA, the purity was 99.3%. We did PXRD and other characterization to make sure that this particular API meets the standard. So these are the kind of salient features of this process. And now we are looking to upscale it at kg scale. At lab, we have done at 50-gram scale. And I think that in 3 batches, it was highly reproducible and we did not face any problem. Thank you.
Can I request you to comment a little bit also on some specific aspects of the value proposition here? You seem to have got very high yield now, more than 90% versus what's
Yes.
reported as 47 or 71%. So
Yeah.
what is the reason that you were able to get this high yield?
So first of all, when we use in the previous processes, they use acetic acid. And it is very, very important to kind of maintain the quantity of acetic acid and little bit. And also, I think we did in 3 batches the previous process, and we found that every time yield varies. Sometimes it goes around 40, sometimes it goes around 50. So reproducibility is a big problem, and every time you see two impurity formation that were consistent in the previous processes. Since in this case we are not using any acid, and also the temperature is not required. So by that way, it actually gave synthocene in more than 90% yield. Also, we tried to look in the possible mechanism, and as was shown in the previous slide, actually the water itself works as activator for the coupling reaction, and hopefully because of that, we got a very high yield.
Yeah, so I think I'll open the floor to a couple of very quick comments from either Dr. Singh or Dr. Chavan, if any, commenting on what they see as interesting in the specific particular technology, and if they have any questions.
I would say this is an excellent condition developed by Dr. Srivastava, performing reaction in water at room temperature, thereby avoiding the impurities found in the previous reaction. I have only one query. He said the addition of alcohol takes place
Yeah,
in the reported room. So here,
yeah,
did he see addition of water? I believe it is adding to the vinyl quinoline.
yeah. Vinyl chlorine, yes, yes. Alcohol was adding to vinyl chlorine. Actually, this particular process, we also used a lot of substrates that we have reported in the publication. And alcohols, thiophenols, all those, and could be added to vinyl. But water addition, we did not observe.
Okay, fine.
I think I just thought I will request Dr. Singh to comment. This process has a lot of green credentials, being
Yeah.
aqueous, run at room temperature, carbon efficiency, E factor. Does it really matter to industry or not? This is for Dr. Singh.
No, yeah, Premnath, you are right. It matters. So I have gone through this. Costing looks attractive. Now, one question to Dr. Srivastav. This, DRL, I think, has entered last year in March 2024 into a license agreement with pharmas. Okay. Now,
Yes.
the chemistry you displayed, just now, is this outside the scope of any patents of pharmas? Have you checked
Yeah, this is
IP?
outside. Yeah, this is outside the scope of format. Yes.
Yeah, so there is no chemistry. Similar chemistry is not patented by pharmas, correct?
No, no. As per my knowledge and
Yeah.
uh, kind of search reports, we did not see that.
And did you also check submarine patents? Because that's very difficult, Many times, I don't know whether you filed a patent, I missed that.
Yeah, we filed a patent and we got a ODIP report. And as per ODIP report, they fish out all the patents that have been filed by Formas and other people regarding this molecule. And as per their report, there was no overlap.
Okay, so my advice is, you monitor for next 18 months, from the date of your filing,
Okay.
so that in the event is there any submarine patent which comes out, and taking the similar chemistry, so please go ahead and do that, for next one
Okay.
year. Okay.
Yeah.
Rest,
Yeah.
Dr. Premnath is all okay. Yeah.
Yeah. I think that's good. There's a question from Dr. Veera Reddy, which basically is about the catalyst. Do you use any catalyst, and if so, what?
No, actually, interestingly, in the beginning we tried heterogeneous catalysis, and then that also worked. But later on, we found when we did the blank reaction, we felt that that is not required, so we did not use any catalyst.
Okay. Dr. Singh, my question on green credentials, is there any specific decarbonization penalty or carbonization penalty for pharma companies now? Because especially with the Europe coming up with all its carbon taxes and so on and so forth.
Uh,
Is
not
there a
yet.
credit? Will there a credit yet for green credentials?
Not yet, but under PLI scheme, those products which are a very, very high volume and, some of the starting material which are coming from China, government is really looking into that. Yeah.
Okay. So I think we are a little late, Dr. Srivastava. So if you don't mind, we'll continue with the next presentation.
Okay.
Right?
Yeah.
So thank you very much. Thanks a lot. Yeah. Over
So
to you,
much.
Pragya. Yeah, thanks.
Thanks,
Thank
Dr.
you. Thank you.
Srivastava. We'll now move on to our next presentation. It is sitagliptin from Dr. Samir Chikkali, CSI Rensil. Requesting my colleagues to kindly share the screen.
Good evening. We are amidst a global diabetes epidemic impacting hundreds of millions of lives and straining healthcare systems around the world. Effective and affordable treatments are crucial. A key weapon in our arsenal against type 2 diabetes is sitagliptin. Sitagliptin is one of the best-selling oral medications that help control blood sugar levels. It belongs to a class of drugs called DPP-4 inhibitors, which work by increasing the body's natural ability to lower blood sugar. It is often used as a monotherapy or a combination therapy with metformin. The global market for sitagliptin is projected to reach a staggering $60 billion by the year 2031. However, current production methods can be costly and complex, limiting wider accessibility and affordability. Sitagliptin was invented by Merck & Co. under the brand name Januvia, of which the basic compound patent has expired in the year 2022. It can be seen that there is a rise of generic manufacturers for sitagliptin since the year 2022, which is precisely the key to unlocking this multi-billion-dollar market opportunity. Existing methods for sitagliptin production can be costly and complex. These methods often involve using a rhodium or ruthenium catalyst with multiple acidic additives. The manufacturer has to bear extra costs due to separate isolation and synthesis of the active catalyst. It can be seen that the latest patent by the inventors are set to expire in the coming few months. NCL's innovative single-step synthesis, which employs a rhodium and ferroline catalyst system, can dramatically simplify sitagliptin production. It can be seen that they can achieve exceptional purity of 99.8% in enantiomeric excess while reducing the number of steps. They have an in situ catalyst generation system which eliminates the cost to separate synthesis and purification. This translates to reduced raw material cost, which are comparable to the commercial values. It can be seen that the sitagliptin which is obtained is significantly higher in yield and purity. The technology is currently at a level of TRL4 and has been demonstrated at 5-gram lab scale. It has been patented with coverage areas in the US and India with a priority date set in 2018. This new method of sitagliptin production proposes the value of a higher enantiomeric excess while getting higher yield compared to the patented process by the original manufacturer Merck. It also has a process for which the catalyst and the ligand system has been patent protected in India and US markets. Overall, it is a very cost-effective process if it's being scaled up to 10 and 100 times its current volume. Who should be interested in sitagliptin production and why? Pharmaceutical companies and API manufacturers who aim to integrate vertically at various steps in hydrogenation as well as in high pressure catalysis can aim to go for this method. This single step process for sitagliptin offers advantages such as a higher yield, lower cost, reduced waste, and simplified purification. The technology is invented by Dr. Samir Chikhli, who's a chief scientist and professor at National Chemical Laboratory, Pune. He leads a group which focuses on polymer and organometallic catalysis. CSIR NCL is a leading research and development organization which is internationally known and recognized for its scientific research in chemical engineering, as well as outstanding track record for industrial research involving partnerships with industry from concept to commercialization. Here is a look at various high-tech, sophisticated infrastructure and analytical facilities at NCL. Here are the industrial collaborations which have been carried out by POMCAT Research Group led by Dr. Samir Chikhli. Looking forward, it is possible to scale up this sitagliptin production with a support from industry partners from a 5-gram scale to a pilot scale using continuous flow. This can be done in partnerships with other CSIR labs as well as other industry partners. The inventor is seeking for licensees for technology and IP on as-is, where-is basis. They seek joint development and sponsored R&D partners to help them scale up and advance this technology. For more information, the contact details are available on your screen. Thank you.
Requesting Dr. Chikhli.
Yeah, very good evening. At the outset, I would like to thank TechX and Dr. Premnath to showcase our technology on sitagliptin. Sitagliptin has been an important drug molecule and has been widely used and has been commercially manufactured. There are patents mainly from Merck, which have been in use, and some of them have been expired. Some of them will expire soon. So while we were looking at sitagliptin molecule as such, there are multi-step processes, but we looked at only the final step because that's the key step where you can introduce enantiomer selectivity. Therefore, we looked at asymmetric hydrogenation of dehydrositagliptin. That's the molecule on the left-hand side. So we use an in situ process where we mix a metal precursor and a ligand, and a catalyst is generated in situ. How is it generated is shown at the bottom. You can see that L12 is the ligand that we identified, and that ligand coordinates with the rhodium metal where the two phosphorus atoms coordinate. And then this acts as an active catalyst, which then takes part into the asymmetric hydrogenation. This ligand is chiral in nature, and then therefore the chirality is induced into the product when the hydrogenation is taking place. So we basically have invented active catalysts wherein the ligand and the metal do the job, and they introduce asymmetry or chirality into the final molecule, which is on the right hand side. That's the sitagliptin. We have done a lot of investigations. We have looked at catalyst loading. We have looked at different types of ligands. And finally, we came to this L12, which gives us more than 99.8% enantiomer selectivity in the asymmetric hydrogenation step. That's the step shown on the top. Then we have looked at how do we separate this. We have also made a salt. We have made a phosphate salt from that. The whole process workflow is on the right hand side. So we start with the dehydrositagliptin. We mix it. We can make a slurry. We then do hydrogenation at a pressure of about 10 to 30 bar in a temperature range of 35 to 90 degree, and the time is about 70 to 24 hours. The asymmetric hydrogenation takes place, then you can reuse the gas when you vent it or collect and reuse it later. Then we can separate the solution, then purify it using a neutral alumina bed, and then we can dry the final sitagliptin molecule. From that, you can make a salt, which we made a phosphate salt from that. So overall, this is the final step of sitagliptin, where you get the active compound, which is R-sitagliptin in our case, and we can do it using asymmetric hydrogenation. This is what I have to say, Dr. Premnath.
Yes, thank you. That helps. I just thought there are quite a few questions coming up. So before that, I thought I'll just request our advisors to comment. Dr. Singh, did you want to comment, or Dr. Chavan, you want to comment? Please go ahead.
Oh, very interesting work and very important molecule, as he himself said. You name any company is not working for sitagliptin in India. I know more than 20 companies, they are involved in developing, and they filed, there are multiple filers for the US FDA. Okay. Now, the most important, the driver for this is the cost. Okay. And I can tell you, even for regulated and non-regulated market, the cost expectation is close to the fully loaded cost I'm telling, is close to $100 to $130. That's what you're talking about is the raw material cost. Okay. Now, second, of course, is the IP. I understand, you yourself said, there's a mark patent, so they cover ruthenium. I don't remember exactly. So whether your catalyst, your process is outside the purview of that mark patent, some of these questions, and the rest, everything is okay. Premnath, go ahead.
Yeah, so we have looked at, so if you can go back to the patent, I don't know, can you change the slide to the patent? Yeah, so we have looked at the patents, both the mark patents, and so we can state we have also done some analysis where our patents is overlapping. So we found that it doesn't overlap. We have the rights, so we have secured the patent for the catalyst as well as for the process, and we have both granted patents in India as well as in the US.
Oh, perfect. Very nice.
Yeah, I just thought I'll also just comment. I think you mentioned, Samir, there are still quite a few questions here which are relating to cost. So I thought you can point that out. That when you do the experiment at your scale, at 5 gram scale, right, you end up using a lot more catalyst, which can be reduced significantly when you scale up, right? So even with those considerations, the number is in ballpark in the right range. So you do expect that the cost will come down significantly, even as you scale up, isn't it?
Yeah, so as of now, I am limited by the way I can weigh it. For example, for the small scale reactions that we do in the lab, the minimum accuracy weight I can take is 1 milligram of the rhodium. So I cannot take less than 1 milligram because of the weighing limitations, or 1 milligram is such a small scale, even if some wind comes, the weight will vary. So we have limited ourselves. We can go down in catalyst loading, but then I have to scale up the reaction. Maybe if I do it on 1 kg or something like that, roughly I can go from, let's say, 1 mole percent to 0.1 mole percent. And if I go to 1 ton, probably I can go to even lower catalyst loading. This, in my opinion, will also bring down the cost.
Yeah. I'll also request Vedant to look up what is the cost contribution of the catalyst per kg of the API. But in the meantime, maybe we can take up a few more questions. Umesh, Dr. Umesh has a question which I'll take up one by one. I'll just leave out the cost contribution of catalyst per kg. That number we have calculated internally, but these are all estimates based on the best numbers you could get on commercial prices. Of course, even that, if it's ballpark, right, it's I think we are almost there. But that said, is the catalyst commercially available? That's a question that's being asked.
So, Premnath, do you want me to answer this?
Yeah, please. Yeah, Samir,
Okay.
please take up the question.
Yeah. So the catalyst, like I said, is in situ prepared. So it is not that there is one component. This catalyst has two components, and both of them are commercially available. So the rhodium precursor that you see on this arrow, that's rhodium cord OTF, is commercially available, as well as the ferrolen ligand is available. So this together in the reaction itself generate an active catalyst.
Yeah. The next question is the chemistry free from IP? I think you've answered that, which you have looked at the other patents and you said it's free, right? And you have also filed your patents as well. The other, and in that, I think, if do you have any more comments on the IP side of it? Or Vidula, did you want to comment anything? Vidula?
Yes, sir.
Do you have any comments on the IP for sitagliptin?
Yes, I'll just so
If you want me to, okay, in the meantime, I can take a few more questions. If it's,
yes. So
okay.
I'll just
The next question is, at what max scale have you taken the batches in the laboratory? And of course, there's one more question on cost.
So
Go ahead,
we have
Samir.
done it on, we have done it on maximum 5 gram scale. We have not scaled it up further.
Yeah. There is also a question which relates to the RMC. Basically, current market RMC cost is USD 90. Whether this process can achieve this market price. I just want to check up. Umesh, is it RMC cost of the API? And is it from which process is this, please? Maybe you can comment on the chat box. So we have not calculated the cost of the biocatalytic process. There's another question which is there, and if that cost is something which you're not able to compare it with at this time, we will need to understand the real cost of that process in a little more detail. Can I just ask one more question, Samir, which is that the enantiomeric excess that you're able to get is very high. That is one of your USPs. what do you think makes it possible? What is it that you do differently that makes it possible?
So this is traditionally well understood. So the ligand that I have shown, so if you look at C1 on the screen, this ligand coordinate in a particular geometry to the metal. Okay. Now, when the substrate is coming, the dehydroxytryptophan is coming towards the metal for coordination and then to hydrogenation. The approach of the substrate is then controlled by the cavity that is generated around the metal. That leads to this high enantiomeric excess.
Okay. There is also, I think, a question. Well, there's a generic question: is it commercially viable? Yeah. So we can only give you the RMC cost at this time to that question. It's for I think industry has to compare it with the options available to them in terms of what processes are available. Sometimes if licensable processes are available, you'll compare it with those or alternate processes. But these kind of enantiomeric excesses, I think, in a chemical process like this, not too many people have been able to show. That's my understanding of this field as well. Do you have any inputs on impurity profiles or anything about the impurities that you might see?
So we have looked at two of them. We did not do like a complete mapping of impurities, but the ones which we could identify, we have done it, and we found that one is there is a dimer formation. That's one impurity that we have. So this we have looked at, and we have identified the retention times and so on.
Yeah. Okay, Vidula, you want to quickly comment on the patent, and then we'll move on to the next one.
Yeah, I had just typed it out. So, mainly what has been claimed in the CSIR NCL patent is, the nature of the ligand. So that is also totally different, which is not being claimed by any other patents, not even in the Merck patent. So the RH precursor catalyst and also the synthetic process of how this catalyst is formed, the precursor, how it is done, is different than what has been claimed in the granted patent. So that is the main difference why this process stands out to be better than the prior arts which are there.
Okay, I think with that we will move on to the next presentation. Pragnya, over to you.
Yeah. Thanks, Dr.
I think
Chikali. Thanks, Vidula.
try and stay within time as much as
Yeah, we
possible.
still have two more to go, so maybe we can limit the number of questions for the next two and reserve it for the last, I would say.
Yeah. Okay, please.
Next is again from Dr. Panda, Olaparib, requesting Vedang to kindly share the pitch.
Good afternoon. The technology that I am going to talk about today is an improved process for the preparation of Olaparib. The lead inventor is Dr. Gautam Panda, and the organization he represents is CSIR CDRI. Olaparib, as you all know, or Lynparza as it is known, is a PARP inhibitor which is approved for ovarian, breast, pancreatic and prostate cancer. In India, the price range for a 150 mg tablet of Olaparib ranges between 428 to 643, which is unaffordable to most of people. What are the market opportunities for Olaparib? So, Olaparib market was valued at USD 3.5 billion in 23, and the innovator company was Kudos Pharma, which was later acquired by AstraZeneca. It has become generic in 24, and the secondary patents are valid till 2031. There are several licensed manufacturers in India like Zydus, Glenmark, Hetero Healthcare, Apazar, Intas, Dr. Reddy's, Natco, to name a few, and there are generic companies like Zydus Cadila and Natco. The existing process which is used for the synthesis of Olaparib has several drawbacks, namely phosphonate synthesis and Witting reaction, use of a genotoxic hydrazine, use of a costly 2-fluoro-5-formyl benzonitrile, and a disadvantage which is a disadvantage for large scale production and more number of synthetic steps, up to 7 synthetic steps. The improved process from CDRI has several advancements and improvement, namely a phosphonate free synthesis. Olaparib has been synthesized using solvents like DMF and ethanol. There are less soluble impurities which are coming from the Witting reaction earlier used, and a moderate temperature has been required. The estimated cost of raw materials is around USD 3090 per kg of product yield. What is the current status of the technology? The status of the technology is TRL level 4. Lab results have been demonstrated up to 5 to 10 gram scale. They have an Indian patent with a priority date of March 22 and publications around this technology. The value proposition for this method is that there is a higher yield of 51% versus 30 to 46%. Number of steps is lower, 4 versus 7. The estimated raw material cost is comparable to the landed cost, USD 3090 per kg of product synthesized. A higher purity with a lower purification cost, better green credentials in terms of reagents used, atom energy and lower energy use, and the API is included in the PLI 1 scheme. Who should be interested? Pharmaceutical API manufacturers, any new API manufacturers who wish to leverage this PLI scheme, companies who are already manufacturing olaparib and want a better process. Why they should be interested is that there are advantages like cost effectiveness, simple reaction scheme, scalable, and fewer steps are required. The lead inventor is Dr. Gautam Panda, who comes with more than 22 years of biomedical research experience. He has worked on cost-effective synthesis of blockbuster drugs. He is the author of more than 132 publications and an inventor of 10 patents. He has industry collaborations done with Dr. Reddy's, Reliance and NATCO. CSR CDRI has several success stories. It is a premium constituent lab of CSIR under the Department of Scientific and Industrial Research. There are more than 13 new drugs which have been discovered and developed, and several success stories as you can see in this slide. They have various industry collaborations. You can see some big names in the slide which is shown. What are the next steps? So it is possible to scale up with support from industry partners from a 10 gram scale to a pilot scale, or even using continuous flow as if required by the industry in partnership with other CSIR labs. What they are seeking is licensees for the technology and IP on as is where is basis, and they're looking for joint development and sponsored R&D partners or scaling up and advancing the technology. In case you need more information, you can reach out to me at the contact details which are outlined. Thank you.
Requesting Dr. Panda to kindly unmute again and tell us more about the technology.
So thank you again for giving me an opportunity to discuss our lab scale technologies of olaparib in this podium. The innovator company of olaparib is the AstraZeneca. If you see the earlier process, it requires the crucial step is a Wittig reaction, which gives very highly insoluble phosphonium oxide that is very difficult to separate at very large scale. Thus, it is affecting the 12 green principles as has been advised by various committees like atom economy, atom efficiency, process mass intensity, mass productivity, E factor, and an effective mass yield and reaction mass efficiency. So in order to address those green parameters, we have devised a 4-step synthesis of olaparib, which is phosphonate free, which is being used in the earlier AstraZeneca process. In this lab scale technologies, we are synthesizing a very crucial intermediate by utilizing SRN1 reaction mechanism. So that is nothing but substitution, a nucleophilic, a radical, and unimolecular reaction in which the generation of a radical under a seal tube reaction which produces a kind of enolate that again reacts with the bromine and fluorine containing a compound giving rise to the intermediate. That intermediate can be again extrapolated by reacting with the hydrogen to give the olapari. So just for comparison, this is a 4-step versus the 6 to 7-step synthesis of the AstraZeneca process. We have drastically reduced the green parameters of this process, which basically gives a very acceptable estimated cost of $3090 per kg as shown here. This technology has been demonstrated at a 10-gram scale, and we are looking for industry partners as is varies basis. And that's all from my side.
Thank you. I think I will request Dr. Singh and Dr. Jawan to add any comments if they want to at this point. And we'll go to the questions following that.
No,
Please
I
feel
think
free to type your questions.
Yeah, cost and chemistry look okay. Yeah. Request Dr. Chauhan to comment on that.
Yeah, see, I have that crucial step of displacement of bromo. First thing is it is done in a seal tube reaction, and you have a fluoro which is a better living group and more activated as compared to bromo, which is meta with respect to the carbonyl or amide group. So do you see any displacement of fluoro as well as the byproducts? And why it goes at the bromo only?
No, actually, this is a radical type of reaction. We know CA bond is more stable than the CBr. So this reaction extensively works on the bromine substituted. That we have done many times, and that we have seen we get only one product.
No, but your yield is only 65%. What is the rest of it?
Actually, this case, a little bit of starting material left. So that has to be separated.
By column?
By column chromatography.
Okay.
Yeah. Okay, let's go to the questions on the chat box, and then we'll take some more as we go along. The first one, of course, is a comment by Dr. Suhas Tambe about product patent being valid in the US. Our reading is that most of the patents have expired in 2024. Now there are generics in the market. In European Union, I think there is some little extended period protection to a supplementary protection certificate. That's what I understand as of now. But we'll look into it if you have a into your comment as well. There's a question on what is the polymorph? Is it novel? Dr. Panda, question to you.
No, we have not done any work on it.
On the polymorph, right?
No.
And at what scale have you established the chemistry? That's the next question.
10 gram.
10 gram. Okay. And I'll just move on to further questions. Have you done any analysis to see if there are any nitrosamine impurities which are detected or studied in the product?
No, we have characterized one impurity, so that we have characterized, but we have not done in nitrosamine impurity. We could not find.
Okay. You could not find, or you did not characterize?
No, we so far we have isolated only one more impurity, and that has been characterized. And nitrosamine impurity we could not find, because I cannot say that we have characterized. We could not find.
Okay. Okay. There's a question from Harish Chandra Jadhav. Does this process involve metal catalyst, and if yes, how much cost contribution is there to that? I think our team members should also look up that and say roughly what it contributes to. Please go ahead. You can go ahead first, Gautam. Pandaji. Yeah.
We are not using any metal catalyst.
Okay. So is there any catalyst here, and what is it?
From the reaction scheme, as you can see, that these are very commonly found chemicals, and there is no catalyst has been used. Only this is a solvent, a DMF, which is a reagent as well, and sodium tertiary butoxide and hydrogen, and also HBTU and diisopropyl ethylamine. So no metal catalyst has been used.
Yeah. Can I request our team members to just look up this question and give an answer? Which is the cost? Which is the thing that contributes highest to the cost? Is a question
Cost.
from Shashidhar Kumar. I think Vedang or somebody has to look it up amongst all the raw materials. Which one is the one that contributes the largest number to the cost?
So in this case, that bromine and fluorine containing material, so that is the, in AstraZeneca process, that starting material cost nearly 1 kg is 60,000 rupees, and in our case, that alternate starting material, that cost comes around from java.com, that is nearly 1 kg 30,000 rupees.
Can I just
So
request a comment from Vedang? Vedang, you
Yes,
need
sir.
to look up the sheet and tell me quickly.
So
On a
the
per kg basis, which is
Yes,
the
sir.
largest contributor to the cost?
The largest contributor is 5-bromo-2-chlorobenzoic acid, the bromine and fluorine containing reagent. And
The cost after recovery is around 589 rupees
fine.
per 5 grams.
Okay. Yeah. Let's just move on to the next question for the moment. Is there any dye substituted product formed in this first stage? Dr. Panda, is there any dye substituted product formed in the first stage from Rajesh Chaudhary?
No, we have not characterized or you found any dye substituted product. No.
Okay. Dr. Veera Reddy asks if carboxylic acid, did the carboxylic acid give any problems?
No, actually, carboxylic acid the only thing which is required for this reaction. We have tried to do with the ester that gives you a lower yielding. So carboxylic acid only we need. So that did not create any problem.
Look, Suhas Tambe has a question. Hydrazine used at last stage, which is genotoxic carryover, could be crucial. Is that a issue?
And this is a very good question. For AstraZeneca, also the genotoxic hydrogen being used, that we could not remove that genotoxic hydrogen. So that is the only common thing.
Yeah.
Now we could not remove this genotoxic hydrogen at the current moment.
So Dr. Panda, just one suggestion here: you make sure your final product is free of hydrogen. Okay. Final product.
Yeah, actually,
There's no contamination.
we did not do extensive impurity profiling.
Yeah.
We checked that HPLC purity and all, so that comes quite acceptable. But as you can see, that extensive impurity profile needs to be done with this process.
Of course. Okay.
Yeah. Yeah, and typically many of these studies are at a stage where the concept is demonstrated, but
Yeah.
further advancement to get it to a point where all the impurity analysis, purification processes all have to be put in place. But that said, it's better done at a little larger scale, I presume. Okay. Okay.
Yes.
I think there are a couple of just final questions. The final stage HPLC purity by area assay.
By area.
By areas. Okay. And I think that's it. We'll probably move on to the next one. I can see Pragna getting nervous about time. So Pragna, over to you. Thank you very much,
Maybe we
Dr.
can
Panda.
reserve those two questions for the last. Let's have the last pitch for the day, and then we can have a discussion. Thanks.
Okay, thank you.
Thank you.
So we move on to our last pitch for the day. I would request my colleague to share for Eva Kaftor from NCL.
Next technology is from Dr. Amol Kulkarni, CSIR NCL, a process for the synthesis of Eva Kaftor in a continuous flow. Ivacaftor is a medication used to treat cystic fibrosis. It is sold under trade name of Calideco, manufactured by Vertex Pharmaceuticals. The cost of treatment for a patient per year being prescribed 2 tablets per day is very high, 3 lakh US dollars per year. The global Ivacaftor market size was valued at 1.5 billion US dollars in 2023. The innovative product patent has been filed by Vertex Pharmaceuticals and which is expected to expire in 2027 in USA and 2025 in India. Recently, Lupin has received tentative approval from the US FDA for Ivacaftor oral granules. Generic version, no therapeutically equivalent version of Calideco is currently available. On the screen, you can see existing process which is being used for manufacture of Ivacaftor. This process has certain drawbacks. It uses harsh conditions, high temperature reactions carried out in batch mode. It uses large excess of polyphosphoric acid and corrosive phosphoryl chloride. To overcome all these drawbacks, we have an innovative process developed by NCL scientists in a continuous flow. On the screen, you can see the reaction scheme for the continuous flow. It has several advantages. It is a short, mild and suitable for large scale production as well as on the spot synthesis. Involves only one purification step, happens at a lower temperature around 0 degrees Celsius. 1 second ozonolysis time, very quick reaction, solvent exchange happens continuously and the product obtained is of purity 99%. Who should be interested and why in this technology? Manufacturers of Ivacaftor and similar molecules for better economical and high yielding process. API manufacturers, they would have an opportunity to diversify, launch new product, new portfolio with a better process. Companies setting up API plants under PLI government scheme to get financial benefits to boost manufacturing. Current status, currently the technology is at a TRL level 4, demonstrated at 6 gram per day scale. Patent status, patent coverage in India, US, EP all granted, priority date 2016. The value that NCL process brings is safe ozonolysis, quick reaction time of 1 second, a higher yield, more than 60% as compared to the 30% for the batch process. All these would impact the final cost of the product at which it can be launched in the market. Lower OPEX cost, lower CAPEX cost and patent protection already discussed in the previous slide. The process was developed by two scientists in NCL, Dr. Amol Kulkarni and Dr. Reddy. Dr. Amol Kulkarni has various domains of expertise. One of them is continuous flow. He has various awards and honors to his credit like ICC DM Trivedi Lifetime Achievement Award, Shanti Swaroop Bhatnagar Award, Sipla Hamid Process Development Award. He sits on various committees and editorial boards of various journals. He has a strong team with various state-of-the-art facilities for analytical facilities. He has a track record of technology transfer and working with industries. Here are a few success stories with the industries in the past that he has demonstrated. He has done several designs of microreactors and flow reactors which have been licensed to Indian industries, and various other technologies have been licensed to industries. He has worked on over 30 industry projects and consulted for more than 25 industries and trained them in flow synthesis. The scientist is now looking to work closely with industry partners to scale up the process from 6 gram per hour to 30 grams per hour. He is also looking to do the quantitative impurity profiling of Ivacaftor. He is seeking licensees for the technology as is where is basis, joint development and sponsored R&D or scaling up and advancing the technology, sponsored R&D projects for ozonolysis in continuous flow. He is also looking for consulting projects for continuous flow. Dr. Kulkarni also has few other molecules for licensing which he has developed using continuous processes, which you can see on the screen. In case you have further questions, you can contact me on the coordinates given on the screen. Thank you.
Requesting Dr. Amol to kindly speak about his continuous flow technology.
Hello, good evening, everybody. Can you hear me?
Yes, we can hear you,
we
Amol.
can.
Go ahead.
Okay. Thanks, Pradnya. Thanks, Premnath, for giving me an opportunity here to present what Dr. D.S. Reddy and myself we worked on a few years back. The idea was to check whether we can make a continuous process for producing this very expensive drug molecule end to end, primarily because it involves ozonolysis, and conventionally doing this particular reaction step needs minus 78 degrees centigrades for almost more than 24 hours, and because of safety reasons, and followed by that three more reaction steps. And we found that probably that bottleneck can be resolved if we make the process continuous, and we could optimize it that this particular specific step we could reduce to 1 second reaction time. Followed by that, then we could even make the remaining steps also continuous, and we have been able to kind of improve the yield almost double that of what we typically get in the batch. And more importantly, the reaction, although we have shown that we started with ethyl indole 3 acetate, ethyl indole 3 acetate can be synthesized using indole acetic acid, which is really cheap raw material, and subsequently kind of we could optimize it to get high yields of the specific beta ketoester and then quinoline ester, and followed by the ring closing to get the ivacaptor through uh, kind of reaction with HATU as a coupling agent. More importantly, there are only kind of one solvent exchange is what is involved in this entire reaction, which improves the overall yield significantly. So I'll be happy to take more questions. We did not do any impurity profiling at this point of time because we wanted to have an industry partner before we get into more details of this. I'll be happy to take any more questions.
Wanted to first just request Dr. Singh and Dr. Chavan to add any comments.
Yeah, I don't have any comment. Dr. Chauhan, you can please.
Yeah, this is a very nice piece of work where you have accomplished the reaction in flow chemistry. I have only one question is, see, you mentioned the drawbacks of using aniline, but do you see what is the difference in the cost of that quinolone moiety which you generate versus your method? Won't it be easier to take that quinolone which is available commercially and then simply do the amide bond formation? And I think the pricing, maybe because of the low demand of this molecule as compared to rest of the drugs, it may be opportunistic price.
Yeah, I think what you said is right, Dr. Chavan. I think one of the most important things here is the where do you start, and you start at the earlier stages. It's possible for you to kind of start with a relatively cheaper raw materials, because making an ester from an acid is not very expensive and not very critical in this particular step because there are no selectivity issues also. So that's where we thought that we can make a difference here. Uh, the molecule is very specific, and as you said, yes, there might be an opportunity cost, but because of its kind of selective target and efficacy, that's why it was so expensive. So even specifically after COVID, where there was lung fibrosis was seen in many patients, the demand for this particular molecule increased. Although it's a relatively low volume molecule, it is a niche molecule that is required for specific cystic fibrosis basically mitigation.
Thank you.
Yeah.
Amol, I just to ask sort of a good example case to demonstrate the ozonolysis capabilities, right?
Yeah. Yeah.
Especially the pharma industry is worried about usually doing ozonolysis, and you're able to do it safely without much as many concerns in a continuous flow process, right? So, can you
Correct.
comment upon some of the other processes also where ozonolysis has been done in continuous flow and is now commercial or
Yeah. So
licensed to others?
yes, yeah, thanks, Prema. So basically, yeah, there is always a worry that ozonolysis is a very expensive process and or way of doing oxidation, and kind of the ozone generator itself will be the most expensive thing. But kind of we have had similar observations or worries when we started working on another product called azelaic acid. where the cost of product is really not as high as what this particular product costs, and even the total tonnage that is required annually is also not significantly high. But still, kind of people have gone for commercial scale because it's a viable module, and your efficiency and the overall economics fits very well. So with a small reaction time of close to 1 second, your footprint also goes down significantly. And what comes out after the reaction is over is basically you are using uh, kind of ozone. The no ozonides are carried forward, and then you get oxygen out, whichever it's ozone that remains in the reaction mass, it decomposes, you get the oxygen out. And so we at least do not see any safety issues. We have operated our other pilot plants kind of several hundred hours so far.
Yeah. So actually, there are questions around that. So I thought I'll just bring that up. Dr. Harish Chandra Jadhav had asked, the low volume API and call for investment for ozonolizer setup, hence investment costs to become critical. What kind of investments are actually involved when you convert a process to a continuous flow process? And how does it bear up? Why is the industry so interested in it right now? Because a lot of them are trying to do a lot of continuous flow processes. So clearly there must be value in it. You can comment on that.
Yeah, there are few specific reasons why industries would need to move into continuous mode. The first is the safety. Second is your overall footprint of the plant decreases significantly. Third is efficiency in terms of the overall yield, selectivity, and conversion. That is much higher than what you typically get in the batch. So the fourth most important thing is the consistency. So there is no batch-to-batch variation kind of issue here. So far you maintain the operating conditions consistently. You're bound to get always the same kind of conversion, yield, and selectivity. So these are the four, 5 reasons because of which kind of the industries are moving in continuous manufacturing at this point of time, mainly in the pharma industry.
Yeah.
Even if it is
So
low.
anyway,
Yeah.
Yeah, so Dr. Jadhav, of course, also commented that it's a great approach as an alternative synthesis. So let me just go to the next question from Rajesh Chaudhary. How is cost comparison of continuous process against batch processes scalable at a plant level? I mean, you might have addressed this, but you might want to repeat it again.
Yeah, it is quite scalable because your resonance times are small. What is required is to make sure that this is a two-phase reaction between a gas and a liquid, where your ozone concentration is only close to 5 to 6% in the entire oxygen that's flowing. And so basically it becomes a mass transfer limited reaction. Having a continuous flow system, mass transfer limitation is almost eliminated. So your reaction becomes extremely fast. And typically these flow reactors have very high heat transfer area. So although all the ozonolysis reactions are highly exothermic, the generated heat is also removed from the reaction mass. So that makes sure that the required yield and selectivity are obtained without any issues. Obviously, one has to optimize that, but this allows you to make sure that you are consistent in all the ways, and more importantly, your excess ozone, which is typically which we lose in the batch mode, where you have to kind of keep bubbling the ozone for several hours at minus 78. That issue does not come. You have to use only slight amount of excess. So even the capex point of view as well as opex point of view is much more reasonable than having a batch process for such molecules.
Yeah. And did you want to just quote a number or two in terms of comparative timelines or residence times between continuous and a batch process for a given process, given synthesis?
Basically, the numbers it's all about the economics, and that will come from your higher yield, smaller reaction times, and mainly you are saving on utility. So keeping something at minus 78 or minus 60 or minus 40 for several hours versus you are doing something at minus 10 or 0 degrees centigrades in few seconds. So
Yeah, that's a scale that I was trying to
exactly, yes.
point to.
Yeah, that clearly means that your overall volume of the reactor as well as your overall plant size shrinks significantly.
Yeah. There is another question which is Dr. Shashidhar Kumar. Is the process in the process, the first stage of ozonolysis, is this scalable, and have you used an excess ozone indicator? I suppose you have answered some of it, but maybe you should just complete your answer.
Yeah, see, this is a gas liquid reaction, so one has to use, and ozone is only 5 to 6%. So you will have to use some excess of ozone in the reaction mass. And the most important, it's not significant excess. It's probably when we did the mass balance of the gas phase which is coming out and the amount of ozone that we monitored at the outlet, it was not more than 8 to 9% ozone as compared to several times ozone excess is used in the typical batch process. So relatively, it's kind of atom economy point of view, it works really well.
Yeah.
So yeah.
Okay, great. So I think with that we are out of questions and out of time as well. So what I suggest is that, by the way, all these videos and all this would be available for people and will be made available to all the participants as well, so that they can go back and think about it as well. So I'll turn over the floor to Pragnya now. So thank you so much, Amol. And if Pragnya, I think unless there's some really burning questions, you should try and close it as quickly as possible. Yeah.
Yeah.
Thank you,
Thank you, Dr.
Thank you.
Kulkarni. My colleague would share a QR code for the feedback that you can give us. Kindly do that. That this helps to improve our format of these showcases. I think we would stop the session here today. If there are more questions, we will be happy to take those and facilitate discussions with the scientists. EOI links have been posted. We'll be following it up with email with the pitches that were conducted today. And last but not the least, I would like to thank all our advisors who have been guiding us, being critical, and helped us in shortlisting, spent time with us with every technology, and all the technology providers as well who worked tirelessly with our team, giving the inputs, doing cost calculations, patent information, and other team members from my colleague who are not so visible. But yeah, they have helped us in putting together the patent and all those information. Thank you so much. We would close here, and we would be looking forward for expression of interest and more questions from the industry audience, which we had today. Thank you.
Thank you very much.
Thank you.
Thank you. Thank you.